There we go. Hi there, my name is Kieran Dillon, and I'm the Executive Director here at the Cancer Vaccine Institute, where we're developing approaches to ignite the immune system to end cancer. We are very grateful to all of our patients, collaborators, and donors for supporting our ongoing research towards achieving our mission. It's my pleasure to welcome you to the third session of the summer seminar series. This is a new thing for the CVI. I do have an announcement for next week's session that there has been a change. The session for July 5th has now been rescheduled for Tuesday, July 26th from 3 p.m. to 4 p.m. PST. We apologize for this change, but it was necessary due to unexpected changes in clinic schedule for Dr. Gwynn. So housekeeping for today's session, please do keep your microphones on mute for the duration of the presentation. And we will be able to take a few questions at the end. And it's my pleasure to introduce Dr. Denise Cecil, who will speak about preclinical development of vaccines, specifically cancer vaccines here at the Cancer Vaccine Institute. Dr. Cecil is an immunologist and principal scientist here at the CVI. She received her PhD in molecular pathology from the University of California, San Diego, and she oversees our preclinical development program for vaccines at the Cancer Vaccine Institute. So without further ado, Denise, take it away.
Thank you. Let's see, why can't I? There we go. So just a little outline of what we're going to be discussing today, considerations in vaccine design, just kind of a little introduction. And then I'm going to take you through our vaccine design workflow, what we do at the Cancer Vaccine Institute. And then next steps, what do we do after we make that vaccine? So considerations in vaccine design, the delivery system, so what type of vaccine, adjuvants, and I'm going to go through these in more detail in the further slides, and then antigens. So first of all, delivery systems. So vaccines can come in different forms, of course. Very famous right now, of course, is nucleic acid vaccines, DNA and mRNA vaccines. For COVID, the Pfizer and the Moderna vaccine are mRNA vaccines. There's also peptide and protein vaccines, so synthetic peptides, just portions of proteins, and then whole protein vaccines as well. Some groups have tried cancer cells themselves, so kind of just breaking those cancer cells apart and injecting them back in to try to show the immune system the cancer. And also people are using viral vectors. And famous, of course, right now is the Johnson & Johnson vaccine for COVID uses a viral vector. And so there's several different delivery systems. And so we're going to go into, I'll talk a little bit about what we do, what delivery system we use further in the talk. So next is to be considered is adjuvants. And Dr. Deese has kind of alluded to this last week. And I just wanted to share with you a little bit of background of why adjuvants are important in relation to this talk, as well as in relation to vaccine development. And so there's two sides of the immune system, the innate immune system and the adaptive immune system. And in this innate immune system, it's not specific and it's very fast acting. So innate cells can go to the site of infection or injury within minutes. It could be there and start try to clean it up. Important in this talk is the innate cells, antigen presenting cells. So the macrophage or the dendritic cell. The macrophage is an antigen presenting cell, but it also goes around and cleans the site of infection or injury up as well. It kind of just phagocytoses all of the dead debris. Dendritic cell is a professional antigen presenting cell. And now that's important because these antigen presenting cells actually take up antigen or proteins that are going to be shown to the T cell. So here's a picture of an effector T cell. T cells are of two types, generally. A helper T cell, we also call that a CD4 positive T cell or a cytotoxic T cell. That's a CD8 positive T cell. This is a killer T cell. This is what's actually going to kill all the cancer or infected cells that it sees. And then the helper T cell, of course, makes sense. It helps orchestrate the entire immune response. That antigen presenting cell is very important because if there's no antigen presenting cell there to activate the T cell, then the T cell just won't work. So that adjuvant will actually activate an antigen presenting cell. So once you get a vaccine, if you mix a little bit of adjuvant in there with the vaccine, you recruit these antigen presenting cells to that site in order for it to start to kind of jumpstart the immune response, if you will, to activate that T cell. And what's really interesting, there's dozens and dozens of adjuvants out there. And I have 2 or 3 examples here in this this cartoon. One, alum is a very old adjuvant, been around forever. It can activate through a series of different signaling pathways, a type 2 response. And so this is a Th2 response. And that can eventually lead to neutralizing antibodies, which of course are important, as we know, in defending against viruses. Interestingly, other adjuvants, here, an example, IFA, MF59, can actually activate a Th1 response. And so what's really interesting is these adjuvants, even though they're not really specific, can kind of skew the immune system by, showing the antigen presenting cell or the innate immune cells the way to a type 2 response or a type 1 response. And so that's really interesting when you're considering what sort of adjuvant to use in your vaccine. Of course, for cancer, we want to use this type 1 response when we're looking at our adjuvants. Of final consideration for this talk is antigens. And Dr. Denise alluded to this last week as well. This is actually her slide. I stole it. It was a really nice slide where, in a viral response, or when you're trying to, vaccinate against virus, and you're trying to vaccinate against COVID, let's say, what you're vaccinating against. It's the virus, right? The sequence of the virus. They have, all of that information, and, they can make a vaccine toward it. But what do we do for cancer? It's potentially not just one thing. There's people have different proteins that are expressed in their cancer compared to other people potentially. So what exactly do we target in this cancer? And so what research have discovered is that we kind of target these new antigens, these antigens that have become new to ourselves, of course, because if we want to try to target ourselves, that could potentially be problematic due to the advent of autoimmunity, potentially. So if we try to target these antigens that are new in ourselves, then it kind of looks like a virus, if you will. And to do that, we look at mutation-based neoantigens and non-mutant mutation antigens. So mutated antigens kind of look like this. Okay, so when we have a normal cell, your DNA looks normal to your immune system. Your proteins look normal, and then your immune system sees these. So this is, of course, on an antigen-presenting cell, or and the T cells can see this, and it looks normal, and so they don't attack it. But when we're looking at a cancer cell, we get all these point mutations in your DNA. Your protein starts to look funny and different, and then your immune system is able now to see different proteins on your in your antigen-presenting cell, which don't look like it's supposed to. So the T cells will say, "Okay, this is not a cell that I recognize. I can attack it and potentially kill it." And so that is one way you could look at. You can target vaccine, but with a vaccination. Looking at these mutated antigens. Of course, everybody's mutations are different. And so this is one way people look at it, but it is kind of labor intensive because it really is just kind of personalized. Another way to look at this is non-mutated antigens. And so this is kind of looking at the same concept where in cancer, so we look at, this is an example of breast cancer. So this is kind of just a section of cancer. And we look at the normal breast, and we're looking at a cancer antigen HER2 new. There's very little HER2 new in normal breast tissue, but however, in cancer tissue, there's very, very high levels of HER2 new expression here. And so what's different about this is that there's such high expression that the immune system recognizes that as being foreign because it doesn't understand that's not what it looked like before in a normal situation. So you see here in the normal cell, this is antigen presenting cell. Looks like this. However, in a tumor cell, you get all this extra expression and it starts to put it on the surface of the cell to activate the T cells. And the T cells recognize that as foreign and potentially will target that and kill that. And so we've shown that overexpression of proteins is an independent predictor of immunity. And so we're able to show here in this HER2 new protein, these are by immunohistochemical staining. If you have patients with really high levels of HER2 new staining on their tumor, they have really high levels. This is the percent of subjects with a T cell response compared to the lower levels. And so really that overexpression is really of the antigens is really what we want to target. And so this is our vaccine development workflow. And so kind of this is what we go through from discovery all the way to product. And so identification of those cancer proteins to target, and I just mentioned that we identify, we don't look for mutation-based, we look for overexpressed antigens. And then we have to identify parts of the protein that are most likely recognized by the immune system. So if you recall showing the immune system has to see, the T cells have to see the parts of the protein, not the entire protein, to become activated. And I'll go into that in a little bit more detail. And then we need to evaluate that immune response from these proteins, peptides that we are able to identify here to see whether or not they're generating the appropriate immune response, because not all peptides in a protein can generate the same type of immune response. And then we make our vaccine. And then we have to evaluate whether or not that vaccine can induce an immune response in mice. And now this is as associated with toxicity because of just putting in a vaccine into a mouse might not generate the toxicity, but actually inducing an immune response does. And so we need to really have that immune response present in the mouse in order to identify whether or not that immune response is associated with any toxicity. So we really have to kind of coordinate that immune response generation in the mouse as we're going forward in our workflow. And then we would like, of course, to see whether or not that vaccine inhibits tumor growth. So it has some sort of efficacy. So first off, I tried to make this a little bit kind of coordinated here. So you'll see all of the parts of the workflow here up in the upper left corner. So you can kind of see where we are in the workflow as we're going forward. So what sort of methods do we do to identify these overexpressed antigens? And so there's several methods. We could kind of just look in the literature. So what other people have published. We've done this before, and we have a vaccine in phase 2 trials right now where we actually identified all the antigens doing this. And so we spent hours and hours, days and days, lots of people trying to identify which proteins we could look at. Another advantage of this is that you have published data from several groups, several different types of tumors, different conditions of those tumors, people treated, not treated, things like that. Additionally, you also have data on whether or not those patients or the antigens that they're identifying is associated with prognosis. And so that's, of course, an important indicator of if that's a driver protein, you really want to target that one if it's associated with poor prognosis. So that's advantage of literature search. Another method that we have used recently is this genomics analysis. And so basically, again, everything's published. It's great. These we can look at genomic data sets of our own and do our own analysis of those genomic data sets and think about how much overexpression there is. These things can be normalized. So we're able to identify proteins that are overexpressed in the tumor of a patient compared to that normal tissue of that same patient, or whether or not it was near the tumor, et cetera. So we've used that in a recent vaccines that are in preclinical development now. And another way we could also do is proteomics. So genomics, of course, looks at the gene expression, which you hope correlates with the protein expression, but not all the time. And so a different method that we could do is looking actually at proteomics, whether or not that protein is overexpressed. And so we have dabbled in that a little bit at the Cancer Vaccine Institute, but we don't typically use that just because the amount of data are not as robust as in the genomics analysis. So now we need to identify those parts of the protein that are going to be most likely recognized by the immune system. And so how do we do that? So I alluded to before the antigen presenting cell. This is very important. If you don't have the antigen presenting cell, you're not going to activate a T cell. T cells do not see naked protein like antibodies do. T cells have to see protein processed and presented in the context of MHC molecules. And so what's an MHC molecule? So basically this is an antigen presenting cell, and they're doing their job. Macrophages go around and they kind of sample the environment, and they pick up potentially, in this example, a bacteria. That bacteria is lysed in the cell, and all the proteins are degraded. You can say that same thing about an intracellular protein. So this is a virus. All those things are degraded inside an antigen-presenting cell. They are fused with an MHC molecule in an antigen-presenting cell, let's say. And then that goes to the surface. And this example here, antigen-presenting cell is presenting peptide in this red dot to the T cell. And the T cell gets activated, as long as it has the correct other stimulations. And so we need to know what goes here in this little red dot. So a little bit about the antigen or the MHC molecules. There's two MHC molecules, class 1 and class 2. Class 1 is expressed in all nucleated cells. So this is a very important concept of tumor escape. So if the tumor will stop expressing class 1 molecules, and the sentinel cells coming around and looking won't see them, and they won't even notice that they're sick. So that's an important method of tumor escape. The MHC molecules are recognized by killer T cells. And then they use specific MHC molecules. And so that's important to know, because when you're talking about vaccination, you're talking about trying to make a more general vaccine compared to people specific, because there's several people have different MHC molecules. And so potentially a class 1 vaccine would not be able to be used on certain people because they won't be able to recognize the peptides that they were shown. So the reason that this is important is, the way that the MHC molecule is formed here. So it really has to have these 9 amino acids and no more and no less, and it has to be a specific way. And so they're very restricted. So MHC class 2, expressed on all professional and non-professional antigen presenting cells, they're recognized for helper T cells. And these epitopes can be presented by multiple MHC class molecules. And you can notice here, here we have the same 9 epitopes being, in the molecule, but you can have little danglers, right? One side or the other, and you can kind of imagine that, this P1 could, maybe move over here if it has the right, if you're thinking about it in different class 2 molecules. And so it's would be more widely applicable to a bigger population. We can use the same vaccine to a bigger population. And so this, of course, I'm kind of leading toward saying that we, of course, are targeting CD4 T cells, class 2 molecules. And for this reason, as well as trying to, kind of target the helper helper compartment of a T cell response, just, kind of, having that more well-rounded, robust immune response rather than only targeting CD8s. So how do we predict which epitopes are going to go into this class 2 molecule? So we're targeting class 2, trying to activate CD4 T cells. And this right here is an antigen, and an example antigen of one of the in our vaccine. So this is the protein, the entire protein of our antigen. And so we actually just kind of feed this in. It's a computer modeling. So there's several algorithms that we can use. Very smart people out there have discovered which particular areas of this protein would be most likely to bind into this right here. And so we're able to identify all of our vaccine candidates this way. And it kind of spits out this graph. So this kind of graph with lots of spikes on it. And at the very top of each spike is what they're predicting to be the epitope that's going to bind in this area. So now we need to evaluate. So we've been able to predict which epitopes are going to bind. And we do have lots and lots of data on that. This prediction algorithms really are predicting the correct epitopes. Lots and lots of data on that. And so now we need to evaluate the immune response generated in human cells. And so people were able, people donate their blood to us, and we're able to look at peripheral blood mononuclear cells. We call them PBMC, and it contains T cells and other cells of interest, antigen-presenting cells. And basically what we want to do is we want to, we've had our candidate peptides that we're able to identify. And we combine these peptides individually with those those PBMC, and we in culture in the lab, and through a series of several really exciting experiments, we're able to then come up with this right here. This is an ELI spot. So every spot corresponds to a T cell that responded to one of these antigens or one of these peptides. And so basically we're able to just count how many T cells are in there that respond, and that's our number. And we do that for all of these. And basically more is better. So the more T cells that respond, the better that candidate looks to us in a vaccine, because it's really easier to boost an immune response that we originally see just kind of in volunteer donors. So people who just volunteer to give us blood, than trying to generate a new response. And so basically that's kind of how we narrow it down, because a lot of times, we'll come up with, I think we had 84 peptides one time we were trying to do this with our phase 2 trial, our StemVac, our vaccine in a phase 2 trial right now. It has 84 of these that we looked at. And so really we're trying to narrow it down. The more, the better. And so that's how we evaluate to come up with our final candidates. So we do the ELI spot. So we do the ELI spot looking for an anti-cancer response and a pro-cancer response. So the reason we do that is because several years ago now, in 2007, they published this. This is NY-ESO is a melanoma antigen. They were able to show the protein goes from 0 to 180 amino acids. So in this black line here, they're able to show that epitopes in green can induce a regulatory response, along with red and blue can induce this CD8 helper T cell and CD4, or CD8 T cell and a CD4 helper T cell sections as well. And so basically this regulatory response is what we don't want when we're trying to make a cancer vaccine, because that turns everything off. And that's natural. That's what we want. I mean, every protein, especially if it's a cell protein, you don't want that cell protein to generate an immune response, because it's yourself and you're going to potentially get autoimmunity, right? So if you look at this section here, and there's, in amino acid 70, you're going to get CD8 T cell response here if you vaccinated at this area, but you're also going to get a CD4 T regulatory response. And so you're going to turn off any immune response generated here from here. And so basically what we decided, and we've published this several times now, is that if we remove areas, so we're not going to use this, even though it's a good spot for CD8s, we wouldn't use that because we don't want to generate that regulatory response. We don't want to turn anything off. So we would go here, potentially, if we were going to use this. And so basically, every epitope that we would test for a regulatory response, and we look for that by the cytokine IL-10. And we test for a CD4 T cell response, and that cytokine is called interferon gamma. And so we do 2 ELISpots for every epitope that we look at. And we did that here. This is an example. This is in revision right now for our prostate cancer vaccine. We did that. So we had 3 epitopes that induce this regulatory response. We call that Th2, so that type 2 response. And we had 3 epitopes that induced that anti-cancer response. So this is interferon gamma and IL-10. And I'm just showing here that this is in the mouse. This is a ELISpot, again, in the mouse. So we have those 3, and then we vaccinate those into our cancer model. And we measured tumor growth. So we vaccinated before we implanted tumor, and then we let them grow. And you can see here, this is the adjuvant. We have adjuvant alone. That's what we use as our control. And that grows, here to 1,000 millimeters. But if you vaccinate with the type 1 epitopes that we saw here that are only interferon gamma producers, we get this inhibition of tumor growth. If we vaccinate with the blue here, that only induce IL-10, again, that regulatory response that turns off any sort of anti-cancer response, we don't get tumor inhibition. If we combine them here in this gray, we don't get inhibition either. So this type 2 response completely turns off the type 1 response, and we get no inhibition. And so we've shown this again in a different antigen, in a different model, in an ovarian cancer model. And so this is kind of how we go forward, where we're not going to use these type 2 epitopes. We're only going to use the type 1 epitopes. So next we need to construct our vaccine. And so I kind of alluded to earlier, looking at these genetic vaccines, make an mRNA vaccine, for cancer, of course, people are testing that. mRNA is, to make mRNA is a little bit more complicated than just making DNA. And so we use DNA in our vaccination strategy and our delivery method lately. And there's many advantages, both theoretical and practical advantages to using DNA vaccines. There's this intrinsic adjuvant activity because it is a DNA vaccine. So you get kind of, a boost in that innate immune antigen presenting cell activity based on just having your DNA. There's potentially prolonged expression because you have this DNA that can, kind of go into the cells. And we have actually, in one of our trials, looked after 1 year of vaccination, and we were able to still see the plasmid DNA being expressed at 1 year follow-up. There's also the potential to have both a CD4 and a CD8 T cell response due to the inherent the way that it's expressed for in both MHC class 1 and class 2 because it's intracellular. And then there's many practical advantages. Of course, it's relatively inexpensive. It's very easy to make this DNA in bacteria. Bacteria naturally make these. They're called plasmids. It's just a circular piece of DNA. And they can share them between bacteria, and so they're really easy to share and to get out of the bacteria. They're easy to manipulate. Here I'm showing this one has 5 different antigens. We have several vaccines right now that have 4 or 5 different antigens. And you can, transfer these around, and I'm going to discuss that in a minute. So pretty easy to manipulate. We are avoiding viral vectors here. Of course, virus itself is, obviously a good inducer of an immune response. However, some are able to get a response to the virus alone and not potentially the antigens that we're looking at. And so, we kind of try to avoid that right now. There's different technology coming out, though, that potentially we could use virus. They're safe and well tolerated. We have lots and lots of follow-up data, years and years, decades of DNA-based vaccines, and very safe and well tolerated. And of course, they're stable. You can find DNA on, sarcophagus and stuff like that. So it's pretty stable. And so when we're talking about considerations to optimize a multi-antigen vaccine, and so I showed earlier, we have 5 different antigens in one plasmid. So how do we know which one to put where, and what should we put between them? And because now we're generating this, 5 antigen protein that's not natural, right? Because we've chosen specific areas of our antigens that we're going to be in our vaccine. Remember, ones that we only are going to be inducing that type 1 response, that anti-cancer response. And so for this example, of course, we had 5 antigens. There's 120 possible permutations. So the way the antigens can go in order. So basically, we're able to do some sort of computation and analysis and in silico prediction. And then we can look at in vitro protein expression and if that changes. So there has been some data showing that, protein expression can be affected by the way that the proteins are arranged. And then we look at immunogenicity, of course. So we vaccinate the mouse, and then we looked whether or not there's a superior candidate on generating the immune response desired. And so the computational analysis that we look at, we predict the physiochemical properties of the protein. And so this is important because, whether or not there's a difference in stability. So once it's inside the cell, is it stable? Because again, we need to have that protein be expressed. We need to have that protein be then put on MHC molecules. So is that protein stable? Of course, it needs to be soluble, right? And then how long is that protein expressed in the cell? And so these are all things that can be predicted based on, physiochemical properties of that particular protein. We also look at secondary structure. So if you remember from your basic biology, proteins are, they have a primary structure, a secondary structure, and a tertiary structure. The secondary structure is whether or not there's coils, beta-pleated sheets, whether or not there's helices. And so there are some data out there that epitopes in helical regions are more induce a higher immune response if they are in helices rather than in coils. And so we are able to just look at our 120 permutations just on, in silico analysis, looking at that. And we can put that on our list of potential, if it's a high percent in helix. And then, of course, there's algorithms to look at for antigenicity, because there's so many that you can look for antigenicity for the tumor. You can look for in viruses as well. And then we also look for whether or not they're predicted to be immunogenic to T cells. And again, there's lots of studies out there. So there's algorithms continue to change, and we're able to apply all of our permutations to these 4 different properties. So again, we have our 120 permutations, and we did our computation and analysis, and I just have 3 examples, A, B, and C. And you can see I try to color them differently here with different orders. First of all, we looked at in vitro protein expression to see whether or not that could affect, the protein expression was different between any of these. And we have 5 different antigens. This is a Western blot. So this is protein expression after we put this plasmid into a cell line that can express these proteins. And then we were able to just kind of quantify this over here. And there's really no difference, no significant difference. Potentially this one's a little bit smaller, but there's really no difference in general protein expression for these candidates. And so that potentially is not going to be useful in this experiment as, to pick the candidate that we want to move forward with. So the second thing we looked at was whether or not the immune response was generated. It was a different immune response generated. So again, we have our 3 candidates. I put them up here. And again, this is our ELISpot that we go to, measuring immune response. We can do that in the mouse just as well as we can in the human. So after vaccination, we're able to analyze the T cell response for each of these candidates. So clearly, you can see that candidate A, we did 2 different doses here, was able to induce the significant immune response higher than the rest of the candidates. And moreover, that this candidate could also 50% of the mice that received this vaccine were able to induce an immune response to all antigens. And this is something we don't typically see. It's usually kind of here or there. And then 75% of the mice were able to respond to 4 or 5. So this clearly is a very superior candidate at this dose. So dose is important, of course, compared to the rest of the candidates and the doses. So moving forward, we take this candidate A at this dose, moving forward to the rest of our experiments. So next, we need to look at toxicity in relation to this immune response. So we generated this immune response, and then we go ahead and look at different parameters of toxicity. And so we look for whether or not we generate an acute response. So that's kind of was a week after our last vaccine. So we give 4 doses of vaccine. And then we look for an immune response, or we look for toxicities after 1 week. And then we also look for, we don't do anything, and then we look for toxicities in a chronic state. So that's at least 3 months after the last vaccine. And so we do blood tests, complete blood count. So make sure that there's no problem with any sort of blood cell. And we also do serum chemistries to test kidney function, liver function, etc. And then we also look for pathology. And so we have veterinary pathologists come in and do. We look for, as we're sacrificing the animal, to look for toxicities to say whether or not there's any sort of gross pathology visible at that time to comment from the vaccine. And then, of course, we look at histopathology of all the relevant organs, of course, the organ that we're trying to target. So breast cancer, we would look for the breast, et cetera. And other organs, of course, that could be important, for pathology. And then any sort of autoimmune pathology that we would see that could be potential and potentially problematic. So finally, we're looking at if the vaccine could inhibit tumor growth. So we use, again, as I'd shown earlier when I was trying to make the point about which type of epitopes we should use to generate that right immune response, we use cell lines, so tumor cell lines that are, generated in a mouse as spontaneously, and then they are able to be propagated, immortalized, and you can inject those back in and just kind of measure the tumor. So initial consideration is for your mouse model, you want to make sure that your antigen is being expressed in that mouse model, because if it's not being expressed, then there's no point of trying to use that cell line as to test whether or not your vaccine is working. So this experiment, again, this is in revision, this is the actual data, where we looked at the cell line expresses our antigen. So just the experimental antigen here, we Western blot, we put in our tumor cell line, We put in our positive control, so we know that the antigen is expressed. And then we have a negative, so we know that the antigen is not expressed. And in this experiment, this antigen is expressed in the cell line. However, this one is not. And I can say that we actually did not take this antigen forward in our model. We had a lot of choices, fortunately, for this multi-antigen vaccine for our prostate cancer program. And we were able to, not use this since we had other choices that we could look at, just for ease of looking at whether or not this vaccine is working. And so that's something to consider when you're trying to you're trying to look at all these antigens at once. So finally, this is kind of just a little busy slide, but I'll take you through it, the schematic for immunization. So again, this tumor implant. And so basically what we do is we have programs where we do prophylactic vaccination, so prevention vaccines. So we do 4 vaccines, 14 days apart. And then we challenge with tumor. And basically, we just inject that cell line that expresses our antigen. We inject that under subcutaneously, and we're able to measure, the tumor growth on the mouse's back as it grows or doesn't grow, hopefully, depending on the vaccine. We also have a therapeutic model where we inject the tumor cells first, and then we're able to give vaccine. So we've employed both of those models in several cell lines that we have for our mice. We also have a spontaneous tumor development models that we use. And again, we have a prophylactic as well as a therapeutic model on that. We have spontaneous models that we can generate with a carcinogen. So we have that for colon cancer. We've used that recently. And then, of course, we also have our transgenic models that develop cancer, breast cancer, et cetera. And then we just wait for tumor to grow. So these are different models. This is kind of just data generated from different models that we have. So this is our breast cancer implant model. So again, this is what I was trying to explain. I found this picture showing the tumor growing on the back of the mouse that you can just measure. And then we have tumor volume here as our readout. And this is, of course, control. So mice that just get a sham vaccine or adjuvant alone. And then mice that get our vaccine. And we're able to show that we are inhibiting tumor growth. So we have a vaccine response, and that vaccine response is showing efficacy. We also have an ovarian cancer implant model. And so this kind of uses a different technology because, of course, ovarian cancer is inside and you can't really measure that anywhere, outside the body. And so we're able to use cells that were generated in this model, spontaneous ovarian cancer model that bioluminesce in an imager. And so we're able to show with vaccine, with adjuvant alone, that we have lots of tumor growing inside. And then with our vaccine, we're able to really inhibit that tumor growth. And we measure that by photons per second in our imaging machine. And so that significantly inhibits. And then finally, we have our colon cancer spontaneous model. And so this, again, is a carcinogen that we give to the mouse. And then we sacrifice them at a certain time, and we're able to measure the colon tumors. So these are polyps, kind of similar to what you see here. And we're able to count these throughout the colon and then give, we're showing inhibition here with our colon cancer vaccine. So finally, what do we do after we make this vaccine? So we've completely generated, we've identified, we've validated, we know that it works in the mouse. What do we do next? So this is testing for toxicity. We know, potentially it's not toxic. Then we apply for an investigational new drug application to the FDA, and we manufacture our vaccine, and this can go into the clinical trials, which will be coming up. I guess it was supposed to be coming up next week, but you'll hear clinical trials at the end of the month. So with that, I will go ahead and take questions.
You can either type your questions in the chat or feel free to unmute and ask.
I had a quick question. When you said about phase 2 vaccine that was on trial, I looked at your website. Looks like you have the.
Oh, you just muted yourself. I heard a Th2 vaccine.
Robin, we can't hear you. Oh, great.
I think I'm
So
in a public
some,
network.
oh,
That's
go ahead.
why. Sorry. I was
Go
just
ahead.
saying, like you guys talked about the phase 2 vaccine that you guys were developing. I was looking at your website, and then I see that you guys are developing on the breast cancer HUR2. So whereas the other vaccines are still down the phase, what do you think you guys can are accelerating or doing anything to promote other kind of vaccines relating to other type of cancer?
Yeah, so we have right now we are in very late stage preclinical development on our colon cancer vaccine. So we're just testing toxicity right now. So hope that should potentially be coming quite soon. Let's see. The others, might be several years away because we're just now, doing, very early preclinical testing. But we do have our colon cancer vaccine coming up very shortly.
Thank you.
Thank you. I see that Ken said he was part of the Protocol 109 in 2006. Thank you so much. We really appreciate. Oh, sorry. It says Ken. Hi, Anita. Sorry about that. Thank you so much for joining here and for being in our trial as well.
Hi, I have a question. Great talk. I was wondering, so you're testing efficacy of controlling tumor growth in the mouse models that presumably you're injecting, mouse cancer cells. You screen if those cells express the peptide antigen that you're going to target. But do those mouse tumor cells commonly overexpress the same types of antigens that you'll find in humans?
Yeah, so that's a very good question. That's another potential way that we could narrow down the candidate antigens, that we do kind of a initial screen with our candidates to see which ones are overexpressed in the mouse cell line that we're going to use as well, or the mouse tumors. We've done that, the spontaneous tumors or the implant cell line.
Okay, great, thanks.
Thank you.
There's a question in the chat. Do you think we primarily use genomics or proteomics analysis in this field?
So
context of developing or identifying antigens.
Yeah, so I think they're both useful. So we have I'm just going through thinking about, we've used genomics more often, and we just started using proteomics, but the genomics, I like the genomics aspect of it at the moment, just because there's more data sets. And, we're looking at RNA microarray data sets, and those are published in, just publicly available. And so we're able to combine those data together because, the RNA microarray data chips don't have every, they're from the different companies. They, potentially don't have all of the protein or all of the genes on every chip. And so it's kind of nice to be able to combine those. And then we are able to, come up with a rubric depending on how many data sets we're able that are actually good quality data sets to, if this antigen is expressed in, 75% of all of the data sets that we look at, that is a good antigen because it keeps coming up, right? So that's a good one across several studies and hundreds of people, in different studies. And so we were more confident on that. The proteomics is just not as widely published, used, and we have done it ourselves. But again, I like the fact that we can do, multiple multiple studies from several groups with genomics right now.
I have another question.
Sure.
So I know that you're focusing on MHC2 presentation to activate CD4s. I was wondering if you had done any with MHC1 to CD8s and maybe how those compared in efficacy, or if you never took them that far just due to wanting to have a more like universal cancer vaccine.
Yeah, so we don't start off looking at class 1. We identify all of our epitopes for class 2, and then we kind of go back and say, oh, is there a class 1? If there is, great. If there isn't, well, great, right? But we are, especially in our phase 2 trials right now, we've had some data showing that we are able to generate CD8 function with our vaccine. And so there are some patients that are able to process that into class 1, regardless of us not, generating the vaccine to be, specifically for class 1.
Any other questions? Okay, well, in that case, thank you all for joining today. Thank you, Dr. Cecil. Please do join us on July 14th with Dr. John Liao, who will talk about vaccines targeting gynecological cancers. And a reminder, next week's session has been moved to July 26th, where we'll talk about breast cancer vaccines. Thank you very much.